And I will get into the introduction of our second speaker, Dr. Brian Hunt. So Dr. Hunt is a biological oceanographer at the University of British Columbia. He's also one of my professors this term, and his research focuses on pelagic marine ecosystems and plankton. So he's going to be talking to us today about the oceanography of the Salish Sea and plankton. And he's actually been doing this for, I believe, the past 4 years, if I'm correct, Deb. So yeah, he's been part of this program for a long time. So yeah, thank you so much, Brian, for coming back this year to do another presentation.
Thank you. Yeah, very happy to be back. And yeah, and I think one of the things with going online is having the opportunity for people to zoom in, and I see that we have quite a nice turnout tonight, which is fantastic to see. So I guess I will get straight into it. So I'm going to share my screen here. Are you seeing the presenter mode?
Yep.
Great. Okay. I'm just going to do one other thing. Maybe I can't do that. Okay, that's right. No problem. Okay, so yeah, so tonight I am going to tell you a little bit about the oceanography of the Salish Sea. And I really do mean that just a little bit. So it's just an introduction, because there's so much to learn about this incredible inland sea. And this, what you're looking at here is a picture looking down from, you can see the bottom of Haida Gwaii over here. So looking to the south, and here's the Salish Sea inside of Vancouver Island. I always like this view of the coast, because one of the things you can see very nicely is all of the fjords cutting into our coastline, which are such an important feature of this area. So what we're going to cover today is how the Salish Sea works with regards to the physics, and so particularly looking at things like the bathymetry, the water sources, circulation, and then the seasonal cycle. And all of those are really the things that define the life support system of the Salish Sea, the biology. So the plankton ecosystems, and I'm going to talk a little bit about why they are so important. And then right at the end, I'll just touch on some of the key issues with regards to the changing ocean and how these might be affecting the life support systems of the Salish Sea. Okay, so I'm going to start off here with some bathymetry. And so this is just to take a step back. So the Salish Sea is a very complicated environment. And so I'm going to try here to bring together the various important pieces. And one of them is the bathymetry, because that determines some of the properties of the Salish Sea. So what this map is showing you is, or the color in this map is showing you the bathymetry. So the darker the blue, the deeper it is. And so what you can see, I just want to check, can you see my pointer quite well? You can, okay. So yeah, you can see the darker coloration. So there's a deep central basin. There are these nice deep fjords that run off towards the mainland coast. And then we have various areas where there's shallow bathymetry, like through the Gulf Islands, and then the eastern part of the Juan de Fuca Strait, and then the Juan de Fuca Strait that's running out into the west coast. And then up in the north, we have Discovery Islands, and then Discovery Passage and Johnston Strait heading off to the north. So we have a deep basin. We have some narrow passages that connect to the open ocean. There's a lot of river inputs. The chief amongst those is the Fraser River, but many other river inputs as well. And then we also have the connection to the ocean. Okay, so the primary connection is Juan de Fuca Strait. So if we take a look at the currents, you can see that these are also quite variable depending on where you are. And so in this figure, the blue coloration is low currents and the red coloration is strong currents. And so the things that really stand out here are, generally we have low currents in the main basin. And then as you get into the Gulf Islands and Juan de Fuca Strait, you have really strong currents. And when you travel, you'll see this yourself when you travel on the ferry between Vancouver and Schwartz Bay. When you go through active pass, you can see those strong currents. You can really see it moving there. The water's moving through, and those currents are mostly driven by the tidal action. And so this is very important because it affects the physics, like the water properties that you find in these different areas. If you look right up in the north here, so we have Discovery Passage into Johnson Strait. Those are also areas where you have very strong currents. But you can see that they're obviously much narrower passages. So the connection to the ocean there is much weaker. So the strong part of the connection between the Strait of Georgia, the Salish Sea, and the open ocean is through Juan de Fuca Strait. Okay, so we've talked about gone over the bathymetry and looked at the currents. And now we're going to bring in the water masses. So obviously there's seawater everywhere, but there's a lot of fresh water that comes into the Straits. And the big freshwater event that everyone is probably aware of is the Fraser River Plume. The freshet that happens in the summertime. You have this huge amount of fresh water that comes running out of the mountains from all the way from the Rockies out into the Salish Sea. And that makes this really remarkable plume that you can see from the city. And if you travel across, again, if you travel across the Straits on the ferry, you'll see it quite nicely. This very clear demarcation between that brown, fresh river water and then the blue salty water. And the reason why that brown, fresh river water is on top is because it's not salty. It's less dense than seawater, so it tends to float on the top. And I'm going to come back to that in a little bit. So freshwater, the Fraser River is obviously not the only source of fresh water, but it's important that the Fraser River freshwater comes out in the summertime, as do all the other snowpack-driven watersheds. They all deliver their freshwater in the summertime when the snow melts. But we have a huge amount of fresh water that's being delivered through rain in the wintertime. And so there's a lot of small rivers that also put fresh water into the Salish Sea. And sometimes we have extreme events like last year when we had the atmospheric river in October. We basically had a full freshet, like there was that much fresh water that came out of the Fraser River. So this is very important because it affects the circulation and the chemistry of the Salish Sea. Right, and the reason why we have so much rain is because we are in the North Pacific coastal temperate rainforest, and so we get less rain here than further up the coast. Central Coast is wetter still, but yeah, there's a lot of rain, there's a lot of fresh water, and this is a very important characteristic of where we live. Many parts of the world don't have this much rain, and so the coastal oceans are very different. Okay, so we have water. So we're talking about water sources. We have fresh water that's coming off the land from all over, not just the mainland side, obviously, but there's all this fresh water that's coming out. And then we also have salt water coming in. So there's deep salty water that comes in from the North Pacific Ocean, and it is cold and salty, and because of that, it's very heavy. So it's dense. So it comes in low. It comes in along the bottom. And it has a lot of nutrients. So that deep water that comes into the Salish Sea, into the Strait of Georgia, is very important because it resupplies nutrients to to our inland sea, and that allows for high production. Like lots of plants can grow. Basically, little algae can grow because of all those nutrients. Not just algae, the other macro, like not microalgae, macroalgae as well, seagrasses, those sorts of things. So where does the deep water come from? It comes from coastal upwelling primarily. So what is that? So this is like, now we're getting into some real oceanography here. We're talking about upwelling and downwelling in the coastal environments. And so basically what's happening is that, so in the wintertime, okay, well, let's start at the top here. In the summertime, we have winds that are blowing from the north, and they blow down the coast, obviously. And because in the northern hemisphere, the Coriolis causes water that's being transported to bend to the right. And so that means that the wind blowing from the north, blowing water down the coast, causes a current that actually goes offshore. And because that water, the surface water is what's being influenced by the wind, the surface water gets pushed away, something has to come in and replace it, and that's the deep water. So the deep water comes up off the continental shelf, and it comes up to the surface on the west coast of Vancouver Island. And some of that deep water will also find its way through Juan de Fuca Strait into the Strait of Georgia. Now in the wintertime, the opposite happens. We have winds from the south, and so the winds are coming up the coast. And remember, the water in the northern hemisphere, the surface water moves to the right, so it tends to move into the coast, and we call that downwelling. So it moves into the coast and it sort of pushes down and pushes water off the shelf, the deep water off the shelf. So this is a very important seasonal process. So you go from an upwelling environment in the summertime to a downwelling environment in the wintertime, and the upwelling is very important because it brings new nutrients into the coast, and that supports the growth of all the little marine plants. Okay, and so I wanted to talk about, I just wanted to highlight density and how it works. And I think that a lot of you probably know this already, and probably most of you have experienced this. So like when you go, when you swim in it could be English Bay, it could be in a lake in the summertime, but you swim out there and it's nice and warm on the surface, and you dang your legs down and it's pretty cold, like 1 or 2 meters down. That's called stratification. And so you have the warm water on the top, and the warm water is less dense, so it tends to float there. And you have the same effect with salty water. So the saltier water will be, the fresher water will be on top and the salty water will be underneath. And so you can combine those two together. So like fresh, warm water would be the lightest, and cold, salty water would be the heaviest. I'm just going to play this short video to show you. I'm going to turn the sound off so I can narrate it. I'm just going to hustle along to here. Okay, so what you're seeing here is this is a container where you've got warm water separated from cold water by a perspex sheet that's going to be lifted up now. So the warm water is, there's red food coloring in there, and the cold water has got blue food coloring in it. So you'll see what happens when you allow them to come together. So you can see that the warm water is settling on top of the cold water. And is he going to do that again? No, let's leave that. So, so remember now that you will have I'm going to skip on to the next slide here. So when you have fresh water that comes out into the straits, it will float on top. And when you have this cold, salty water coming in from the ocean, it comes in on the bottom. There's also, these are our main inputs. So we have fresh water coming off the land, we have this cold, deep water coming in, and if something's coming in, something else has to go out, like to keep the balance. And so we have warm, less salty water going out on the surface of Juan de Fuca Strait. So that's going to be water that's influenced by both the deep water that's coming in and the fresh water that's coming in from the coast. They're mixing together, and in the summertime in particular, it's warming up, and that warm, less salty, and so influenced by fresh water, that warm, less salty seawater is flowing out on the surface of Juan de Fuca Strait. So that sets up the circulation for the system. And there's a little bit of exchange through Juan de Fuca, through Discovery Passage and Johnson Strait, but it's far less than through Juan de Fuca Strait. Okay, so how do we measure ocean properties? Well, we use a device like this. This is a CTD, a conductivity temperature depth profiler, and that conductivity is how we estimate the salinity, so the salt content of the water. And we can lower this down through the water column, and it measures at really high frequency, 6 times per second. So you get a very high resolution profile of the water column. And you can measure from the surface all the way down to the bottom. And you'll have, you might have 5 to 6 measurements per meter all the way down through the water column. And then we can use that to reconstruct what the water column looks like in terms of these different properties that we're measuring. So I use water column a lot. So that's a proper oceanography term, and maybe not everyone is familiar with that, but basically we're just talking, the water column is what we're talking about from the surface to the deep, to the bottom of the Salish Sea. That's the water column. Okay, so this is, I'm not going to show you a lot of oceanography plots, but this is one of them. And so this is showing you the temperature profiles for the Salish Sea. And based on the temperature, we can see this is in the summertime. And so what I'll walk you through this. So you can see, if you look at the little map on the top here, we have this red line, which we call a transect, where the ship has basically moved past from the outside of Runoff Strait through Runoff Strait and then through the Strait of Georgia all the way up to Quadra Island. And then along that transect, along that line, there's been CTDs profiling. So we've been setting them down to the bottom. And so from that, then we can reconstruct the temperature profile of the water column. And so here is that temperature profile mapped out from the start of the transect on the left-hand side to the end of the transect on the right-hand side. So this is Runoff Strait on this side, and this is the northern Strait of Georgia, Quadra Island on the right-hand side. And so, and then the gray color shading on the bottom is actually the seafloor. So you can see the shallowest point around the Gulf Islands here is around 125 meters. And the deepest point in the Strait of Georgia here is around 370 meters. And so the coloration is showing you the temperature, and the pinkish is cold water and red is hot water. Well, let's say warm water, it's not quite hot. And so you can see here this pink tongue of cold water that's creeping up through Runoff Strait. Now that's the deep water. That's the upwelled water that's coming into the Salish Sea from the North Pacific. And then on the top here, we can see the warm water in the main, the red coloration in the main Strait of Georgia basin. Okay, so we have the cold salty water that comes in from the Pacific. We have mixing that happens around the Gulf Islands. It's shallow there, so the water is moving back and forwards, and because it's going from two deep locations, it means it's being compressed over the shallow water, and it causes turbulence. So you have turbulence going back and forwards, it mixes up the water, and that's why it sort of homogenizes and ends up more of a sort of a green coloration all the way through by the time you get to the Strait of Georgia. And then we have this warm and fresh surface water in the summertime. And we call this, this is the stratification I was talking about, where you get a heat trapped at the surface and it forms a nice layer of warm water. And the warmest water that you'll find is up in Desolation Sound, not far from here in the northern Strait of Georgia. If you look up on the map, it's this area right on the eastern side. And that's the warmest water in British Columbia. It could be quite a bit over 20 degrees Celsius on the surface there. Okay, so that's what the water looks like in the summertime. So now let's take a look at how the water column changes over the season. So this plot here, so this is, let's just pick one place in the Salish Sea. So we go out and let's say there's a marker that's out between 5 miles off Vancouver, out in the middle of the strait. So we go back there and we sample that every week for the whole year. So this is what this plot is showing. It's showing you temperature profiles from one point over the whole year, from the 1st of January on the left-hand side to the 31st of December on the right-hand side. This is actually at Nanu Station, which is up closer to Nanaimo. And so the coloration is showing you temperature again. So the pink is cold and the red is warm. And so you can see in the wintertime, when the sun is low, we have cold waters, so there's a lot of cooling going on. And then in the springtime, the surface starts to heat up. And then we have the stratification, this warm surface water through the spring to the fall. And then it starts cooling down again. And you can see that we start getting the winter cold around end of November time. It's starting to get really cold again. And some of that cold water, you can see here, finds its way also into the deep waters of the Salish Sea, and that is what contributes to the cooling, the seasonal cooling in the deeper layers as well. Okay, so there's a temperature change, and there's also a change in the sunlight, and that's very important because that affects the growth of the plants in the Salish Sea. So now we're going to move away from the physics, and we're going to go across to the biology here. So why does oceanography matter to the food web? Here's a very simplified food web. We have sunlight that supports the growth of the phytoplankton, so the little microalgae, and they're consumed by the zooplankton, and then the zooplankton are feeding small fish like herring, and then the herring are being consumed by various other species, and ultimately all finding their way to top predators like the marine mammals or some of the seabirds as well. Okay, so the plankton. What are plankton? These are free drifting organisms, and there's two main types. There's the phytoplankton, which are the plants, and the zooplankton, that are the animals. The ones pictured here are all zooplankton. No phytoplankton. Well, there will be some phytoplankton, but it's too small to see in this image. And there are thousands of species. Okay. And so how does the oceanography matter? Well, first of all, there's light. Okay, so let's talk about the physical processes. So the phytoplankton cannot grow without light, and so the seasonal cycle, that winter, the spring to summer sunlight is very important. That's when you get most of the growth happening. This is the growing time of year. There's nutrients as well, and so if you remember, there's a lot of nutrients in that deep water that comes in seasonally from the upwelling. So that's very important, supplying nutrients to the phytoplankton. And then we also have temperature that is very important for the organisms in the Salish Sea. And so these three components are particularly important for the phytoplankton and zooplankton, which are the base of the food web. So without those, we would not have any of the other organisms in the Salish Sea. So I've talked about the seasonal cycle, and I want to just elaborate on this a little bit with like how it is relevant to the plankton, the phytoplankton in particular. And I'll do that with this figure here. So the seasonal cycle is important for two reasons. It affects the sunlight available, how much light is available for the phytoplankton, and it also affects the mixing and nutrient supply to the surface where the phytoplankton need to be to grow. So if you look at the winter time, we have low sun, so there's not much sunlight for growth. There's strong winds in winter, and that means that you get a lot of mixing, deep mixing in the water. And that's indicated here by these white squiggly lines. And it can be, the water can be mixed so deeply that the phytoplankton end up on a routine basis in areas where there's not enough light to grow, especially at this time when light is a very short supply. But the other effects of this mixing is that it brings deep nutrients up to the surface. So it resets the water for the spring growing season. It's like tilling the earth in a way. So you get into spring, now the sun is higher in the sky, there's more light. We put a lot of nutrients in the surface because of the winter mixing. And then we start to get some warming and things stratify a little bit, and the phytoplankton get trapped in this beautiful, it's like for them, it's like this is the best time of year. Trapped near the surface, it's warm, you're getting lots of light because the sun's higher, and you're trapped at the surface as well, so you're not in the deeper, the darker parts of the water. And you have lots of nutrients. And so this is when we have what we call the spring bloom. So phytoplankton grow like mad. And that is very important for lots of animals key in their life cycle to be perfectly timed to coincide with the spring phytoplankton bloom. Now they grow so quickly, they use up the nutrients, and then through the summer, there'll still be relatively high production, but it can depend a little bit on the resupply of nutrients. So like how often, if you have a couple of nice windy days, that will bring some fresh nutrients up to the surface, and then you'll get the phytoplankton growing again. But if you have just hot, sunny conditions the whole summer and very little wind, you'll end up with basically very low phytoplankton biomass because you will not have nutrients supplied to the surface anymore. And that's one of the things that's climate warming, one of the effects that climate warming has on the ocean is that it causes very intense stratification and limits resupply of nutrients, and that so it can actually decrease productivity. Okay, so the Salish Sea is connected to the land and the sea. This connection to the land is through the fresh water, and all of those, all of the nutrients and the chemicals, terrestrial chemistry that is transported to the ocean by fresh water. It is connected to the sea through that upwelling, and this together, these two components determine the circulation and also the nutrient supply in this for the Salish Sea life support system. And it is highly seasonal, and so the sunshine and nutrient supply drive the growth of phytoplankton. And this is a key event which has already been happening this season is the spring phytoplankton bloom. We've already had some really big blooms going on in all parts of the Strait of Georgia. So why do we care about the plankton? So, well, the plankton are the foundation of the food web, and they ultimately provide most of the food that supports the ecosystem. They produce 50% of the oxygen that we breathe, and they remove CO2 from the atmosphere, which combats global warming. And a lot of that's CO2, or the CO2 is turned into biomass, and then a lot of that will find its way to the seafloor where it can be sequestered in sediments. I saw some comments in the chat about seagrass or eelgrass taking up CO2 and so contributing to blue carbon, but the main removal mechanism for CO2 is going to be phytoplankton in the global ocean, and certainly important in the Salish Sea as well. So here we can see our trophic pyramid, and so we have a lot of phytoplankton out at the base, and then the zooplankton, the smaller fish and squid, and then our bigger species that are feeding on those the smaller organisms. And it's drawn like a pyramid because that reflects the biomass distribution. So the base of the pyramid is the biggest. That's the phytoplankton biomass, followed by the zooplankton. So that's most of the organisms in the ocean are these planktonic species. Okay, so here's an image of some colleagues that are busy sampling the plankton. The net there is what we call a bongo net, and you use that to sample the zooplankton. And this is one of the common methods used in our area. And then in the right, the inset here at the bottom, we have some water bottles called Niskin bottles that we can lower over the side, and we can send them to different depths and open, and then we use a device to trigger them to shut, so we can catch water at different depths and look at what's there. And that's what we use to sample the phytoplankton if we're trying to see what species of phytoplankton are there, and also how they're distributed in the water column. And we'll also sample other things from this water. We might measure the nutrients, we might measure parameters relevant to ocean acidification, bacteria, viruses, all kinds of things. Okay, so let's take a look at some of these groups. So this is and we'll start off with the phytoplankton. So here is an image of some microphytoplankton, and so phytoplankton is a broad term, and we might loosely just call that microalgae, but I wanted to really emphasize how diverse phytoplankton really are. And you know, they're small, you can barely see them. Some of them you can see with the naked eye, but they have an enormous size range. And this figure has a nice way to put it all together. So the smallest phytoplankton are these Prochlorococcus, so these little cyanobacteria down on the left-hand side here. And then the biggest phytoplankton are over on the right. And so there's a few different types here. This is just a small representation of what's there. There are thousands of species. But the main point is that they cover a 4 order of magnitude size range. So that means if the smallest phytoplankton was the size of a herring, the biggest phytoplankton would be the size of Manhattan. So that's an enormous range. It's a much bigger size range than you see in the upper trophic levels. And so if you think about it, that must be very important for the animals that eat phytoplankton, right? Like if you're eating something the size of a fish, it's very different to eating something the size of Manhattan. And so although the plankton systems, the plankton, all the plankton, phytoplankton and zooplankton are so small, and you can catch them in a jar, and you think you're seeing what's there, and you're understanding what's going on. It's incredibly complex because of it's such a broad spectrum of animals in terms of taxonomy, like the different species, and then the size ranges. There's all kinds of interactions going on there. Okay, so one of the main groups, one of the spring bloom phytoplankton are mostly diatoms. So diatoms are large phytoplankton. You can see them with the naked eye if you look closely enough. Not all species, but many species. So they often form chains, which can be millimeters long. And they have cell walls made out of silica. So they really depend on high silicate concentrations in the water, which we have in our area because in part because of the glacial runoff. So a lot of the erosion from the mountains and is eroding silicate into the water, and that gets washed out into the Salish Sea. So we have very high concentrations here. So good for diatoms. So they make these beautiful silica cases, and those are there to protect them from predators, because they're not just that, but that's part of the function, is that they're hard and difficult to eat, and a lot of them will have spiny processes to prevent predators getting at them. We also have Dinoflagellates, so it's another important group, also fairly on the larger side, and they're quite interesting because they can be predators, they can be plants, they can have chlorophyll, they can be photosynthesizing, or they can be predators, or they can be both. So they can be what we call mixotrophs. They can be, which means that they are both plants and that produces biomass, and then a consumer that is eating other animals. So there's quite a few different species, and some of them are very important because they are, like Alexandrium is paralytic shellfish, poison inducing, and so there are a few toxic species, and they're also something that I think many of you will be familiar with is this organism. You might not know it as Noctiluca, but take a look at it. It's got this little round, it's like a round sac, and they form blooms like these. Red, some people, you might call them red tides. Red tides are often thought to be toxic, and these are not toxic. They are, but they do make enormous blooms, and you'll see them in the summertime here. During the day, you'll see this red coloration on the sea surface, and then if you go back at night, you could be lucky and see the bioluminescence. So they're very bioluminescent. So when waves are breaking, they'll bioluminesce. Where if they get disturbed, if you walk through the water, they'll bioluminesce. And they can cause problems if you have a very big bloom and then they all die and then sink onto the seafloor. They will decompose, so bacteria will break them down, use up the oxygen, and the oxygen concentrations in the water will get very low, and that can affect organisms in the water. So another interesting group is Coca-Lithophores, and so this is the organism here. So it has these calcite scales, basically, that makes a hard, compact calcium carbonate shell. And every summer, we have big blooms of Coca-Lithophores on the west coast of Vancouver Island. And so NASA very proudly presents pictures of these because they're usually very pretty every year. You can see big eddies and meanders. And in sometimes, actually, the only time I'm aware of it was in 2015 when we had the first big marine heat wave. They were seen in the Strait of Georgia. So I remember seeing them in Howe Sound and also up on the Central Coast, but they were seen in the Strait of Georgia. So that's potentially something that can happen with with increased warming conditions, that we'll see more Coca-Lithophore blooms. And they are responsible for the White Cliffs of Dover. That's all Coca-Lithophores. So they can make enormous biomass and build up sediments. Okay, so let's take a look at the zooplankton. Here we have an array of zooplankton. I want to focus on a few important groups. Copepods. Okay, copepods are the most abundant of the zooplankton species. And there are many, many different species, over 10,000 species. They occur in freshwater as well. Some like this group here, we call this Matridia. They're also very bioluminescent. So when you see those blue sparkles at night, maybe when you're lifting a boat anchor, for instance, it's often these guys. Most of the copepods, the important copepods anyway, are grazers, the ones that are important for fish. So there's a few important species that will eat the diatoms and dinoflagellates, and they're an important link to fish. Another important grazer is the krill, which we call euphorsids, and their Euphorsia pacifica is one of the important species. Very important on our coast. Salmon really like them. Herring love them. Opalescent squid love them. Basically, everything loves to eat krill. And there's actually, not many people know that there's actually a small krill fishery in the Strait of Georgia that operates in a couple of the inlets up in the, like Nice Inlet, for instance. I think the fishery is something like 500 tons a year, and they're collected for feeding to, it's mostly for fish meal. So, which is very interesting. I think the only other krill fisheries I'm aware of are in the Southern Ocean, so it's quite unusual. Pteropods are one of my favorites. These are sea butterflies, and they are a free-swimming snail, which is a little bit bizarre, really. So it has a shell like a snail. You can see it over here very nicely, and then the foot has developed into these two wings. And pteropod means wing foot, so it flies around like, I guess, a bit like Dumbo the elephant. It flaps its wings and it can fly through the water column, and it's a grazer. Fish love to eat them, particularly pink salmon, chum salmon also really like them. And they are important indicators for ocean acidification. So they're considered a sentinel species because they have a calcium carbonate shell. Unlike those Coca-Lithophores that I showed you earlier, which have a calcite shell, these have an aragonite shell. And so aragonite's a different form of calcium carbonate that's more susceptible, less stable. So it dissolves more easily, basically, with ocean acidification. So these are being used to monitor ocean acidification around the world. All right, then we have our jellyfish species. The common ones are the moon jellies, water jellies, and the lion's mane. Jellyfish are quite interesting. One of the things we discovered is that Aurelia in particular, so the moon jellies, they quite like docks. And so the polyps like to grow on the undersurface of structures. So they do particularly well around docks, and you can find enormous blooms. I wouldn't, let's not call them blooms, but like aggregations of jellyfish Aurelias in these locations. And so we've estimated some of these jellyfish smacks, the aggregations, to be up to 100 tons. So just really enormous aggregations. Okay, so that's a few species of plankton, but there's a lot of, those are like typical plankton, but there's a lot of species that you might not expect to live in the zooplankton, but are there at least for part of their lives. We call them meroplankton. They're not there all the time. And so one example would be squid. We have many benthic species, basically all benthic species, the vast majority anyway, have got a planktonic larval stage, including sea urchins and sea stars. And then most species of fish also have a larval planktonic stage, and one of those is the herring. And I'm going to pause on the herring here because herring have a really, they're such important fish on our coast, and they really interact very nicely in a very complex way with the plankton. So this is an adaptation of an ancient picture that was put together by a British researcher for Atlantic herring. But it shows basically the connection of herring at all life stages to the plankton. So as larvae, they're part of the plankton, and they're feeding on very small microplankton, even phytoplankton. And then as they grow, they're feeding on bigger and bigger things up until the adult stage, where you might here in BC, they particularly like to feed on krill. So this is a very complex web of interactions, and when they're larvae, they actually could be preyed on by other zooplankton. So that's what's being indicated by these arrows. So there'll be some predatory species that will eat larval herring, and actually one of the biggest. I'm just going to go back a bit. So these jellyfish, really, water jellies and Aurelia moon jellies are known to feed quite heavily on herring larvae. They can cause up to more than 90% mortality. Okay, all right. So this is a site that I think some of you will be familiar with. This is the top end of Denman Island. This is the herring spawn. So this is a major event on our coast, and so one of the important features, one of the important features ecologically, but one of the things that's really born out of the oceanography of this area, it's a very productive inland sea, and it's able to support this enormous stock of herring, which this is the one stock that's been doing at least fairly well in British Columbia. And the herring is it's just such a nice case study of a species that really interlinks with different components of the food web. So they are consumed at all parts of their life cycle by many different species, including shore species, which might be feeding on the roe or the dead adults, and as well as many top predators. And they're all supported by the plankton. Okay, so the plankton are really underpinning this complex food web. And another species that we can see in here is I have to highlight the salmon. I do have the salmon in my background for my Zoom picture. These are juvenile salmon in the Discovery Islands, and they are also very dependent on the productivity in the Strait of Georgia. And a lot of what our research is about these systems, like how these systems are changing, the food systems are changing that support these species like salmon and herring. Okay, so I'm emphasizing this point over and over again because we care about the plankton food web because they support the food webs that we and other organisms depend on. And so light, nutrients, and temperature, as I said, are very important for the phytoplankton and zooplankton. And that is the reason why plankton food webs are changing. It's because the supply of particularly nutrients and temperature and other factors like ocean acidification are impacting plankton communities. And I'm going to run through these quite quickly, but I'm going to make the main points here. So we have ongoing long-term anthropogenic increase of, or anthropogenic-driven CO2 increase. And this is the atmospheric CO2 at Mauna Loa Observatory. So this is atmospheric CO2, and this gets transferred into the ocean. And that upsets the carbonate chemistry and basically leads to consumption of carbonates, carbonate ions that are dependent on that organisms like pteropods and Coca-Lithophores and others depend on. It also affects the pH of the water column, and that can affect physiological processes for many organisms as well. The most noticeable immediate effect, though, is this dissolution of shelled animals. And this is a problem certainly already in parts of the Salish Sea. That CO2, self-same CO2, is causing atmospheric warming, and so we have a long-term increase in global ocean temperature. So this is from 1880 to 2015, but this is, we have exactly the same time series for the Salish Sea. So although this is a global ocean temperature, you will see the same warming happening in the Salish Sea. So this is about 2 degrees over the last 120 years. And it's not only this long-term sort of steady warming, there's also what we're seeing is an increase in extreme events. So one of those would be the marine heat waves, the first big one we saw in 2014 and 2015, but there's subsequently been 2 others. Atmospheric rivers would also fall into the same category as extreme events. And with the heat waves, what we saw was the introduction of, the visitation at least by several new species, including some phytoplankton species. There was a high prevalence of a group of diatoms called Pseudonychia. which produce a toxin called domoic acid, which is problematic for vertebrates. And so there were some issues with or high mortalities of seabirds and also sea lions. We saw a lot of ocean sunfish. No one was complaining about that. And then we had, very interestingly, we've had these huge outbreaks of tunicates, these gelatinous. These are colonial animals. There's several types of them, and we have been getting various species over the last 6 years now. Every year there are huge outbreaks of these on the west coast of Vancouver Island, and these ones, commonly called sea pickles, you'll see them washed up on the beaches on the west coast of Vancouver Island. And I haven't seen them in the Salish Sea yet, but and which is probably unlikely, but you will see them on the outer coast. So shifts so we see shifts in the planktons and also in productivity, and we're busy currently researching these changes and the knock-on effects that they have on the rest of the ecosystem. So that's one of our important areas of research, trying to understand how what the ecosystem response will be to these changes in the plankton species composition and productivity. And so just quickly, just to highlight a few groups that are working on some of these problems in the Salish Sea. Here we have obviously UBC, Hacker Institute, Pacific Salmon Foundation, Salish Sea Marine Survival Project was an excellent one. Ocean Networks Canada, Fisheries and Oceans Canada, Oceanwise, just to name a few of the many. Actually, one of the things that I find most inspiring about working in the Salish Sea is the huge number of peoples who are passionate about the ocean here and are doing something to make a difference. So we could have an enormous list of contributors to that. And I'm going to stop sharing here, and I think we have some time to take some questions, if there are any.
Yeah, so there's tons of questions. Just before that, I just want to say thank you so much, Brian, for coming back and giving a really interesting presentation as always. And yeah, hopefully you'll come back next year, maybe. And also for anyone who's a UBC student or knows a UBC student, I would highly recommend taking one of Brian's classes. I'm really enjoying it right now. Yeah, so we have the questions. I think Deb and Anne will help moderate those.
Mm-hmm. Yeah, there we are. So that was fabulous. Really, really enjoyed that. And yes, we do have a list of questions. So I'll start with a couple. Deb, you want to come in as well, but one of them I found actually quite interesting, of course, which is glaciers are melting, as we heard from our lovely previous speaker, Sky. So what's that effect going to have, do you feel like, on the Salish Sea? Has anybody been examining that issue?
Yes, we have. I mean, this is something that we're actually thinking about a lot. And I say we, like we have a group. It's like a NOAA-funded group that is looking at the North Pacific temperate coastal rainforest interactions with the ocean. And an important part of that is the snow, ice, rain dynamic, and the loss of glaciers is changing. It has a huge effect on local ocean processes, and because of the scale of our ice fields in British Columbia and Southeast Alaska, it actually, like, the sum effect of that would be quite dramatic. And one of them is shifting the hydrodynamic cycle. So we end up, what we're moving to here is towards more of a winter rain driven sort of circulation in our coast, as opposed to this strong input from the summertime snow and ice melt. So that's one part of it. But then the chemistry of the glacial chemistry is very different to snowpack or rain, and so that's very important. It affects the type of nutrients that run off, and that is going to have an effect on the types of phytoplankton, zooplankton, other plankton species that we have in our coastal waters.
Mm-hmm. Yeah, and part of another question was, what percent is glacier versus rain? Is there any statistics on that? Or
I know, so like this, the statistics that I can share. Okay, like 50% of the rain, 50% of the freshwater runoff comes from small systems with winter rain. Okay, and then there'll be some percentage of like winter rain coming out of the bigger systems like the Fraser, Skeena, Nass, for instance. But then the rest of it will be snow and ice. And I think it's, it's probably less than 25%. Certainly there would be glacial. Yeah.
Okay. All right.
Yeah.
Fascinating. Cool. Thank you. Shall I keep going, Deb? Do you have anything else you wanted to? Got to mute. Yeah.
There's a question here from Murray. Are there any situations where the cold, salty, deep water would be able to move past the Gulf Islands into the Strait of Georgia? Or does mixing between Gulf Islands always prevent that?
It does come in, but it just comes in modified. So it gets mixed into that outflowing water. And so it, like the nutrient, the good stuff, the nutrients find their way into the Salish Sea. But yeah, it's certainly, sorry about the background noise if you're hearing that. It's cool. It's warmed up because of mixing with the surface water. So yeah, like I say, it's modified, and that's different to other places. Like if you go to on the coast, if you go up onto the central coast and you go into some of the fjords there, when the upwelling happens, you actually see that cold water coming all the way into those fjords, and so they tend to be actually cooler in the deep water than we have here in the Salish Sea, because here it's mixed up with that warmer water, which is interesting because you might have a subtly different like the climate effect might be different in those two, the warming effect might be different in those two locations.
Yeah, great. Here's a fun question. Why does the phytoplankton sparkle? What's the reason?
The answers are often speculative, but I think they're mostly defense mechanism. Yeah. So sorry, go ahead.
Just wondering. So then, what does it like blind prey or how? What?
Yes.
Oh yeah.
So the things that are eating them would be zooplankton, and zooplankton use bioluminescence a lot. They're definitely sensing lights. I mean, they modify their behavior based on lights, and they bioluminesce themselves for predation defense. And so yeah, I think it's a blinding flash that can maybe disorientate the predator, and that would be the same thing that those zooplankton might do against the predator on themselves. Yeah.
Very cool.
Yeah.
Thank you. Bob asked, which nutrients are delivered by deep waters from offshore upwelling versus nutrients delivered by rivers?
Oh, that's such a good question. So the nutrients from we have a pie here.
Erin Ann. Thank you.
Okay.
That looked really good. So the deep water will bring in the things that we measure as important are nitrates, silicates, and phosphates. And then from freshwater, we get from the glacial systems, you'll have a lot of silicates. But like the nitrogen content of the freshwater systems is very low, particularly the rain-driven systems. And so they actually, if you have a lot of rain, you can dilute the nitrogen to the extent that it becomes limiting for phytoplankton. But they have a lot of organic carbon. We see that brown water in the freshwater. That brown, it's been leaching a lot of, organic matter. So that runs out into the coast. So that's mostly organic carbon. And that is very yummy stuff for bacteria. So the bacteria really enjoy that. And then the other thing that's key is iron. So iron is very important for phytoplankton growth and needed to photosynthesize. So that's why if you go out into the open ocean, into the Gulf of Alaska, there's lots of nutrients there, but it's not very productive because there's not a lot of iron. It's far from land. And so iron actually then becomes limiting. And so if you can get iron out there, so people talked about, iron fertilization. If you can put iron out there, you can, things will grow. And so, yeah, iron from freshwater is very important. And actually things that, I don't know if people know about Haida eddies, these big eddies that form in the springtime off the southern end of Haida Gwaii. these get transported out into the Gulf of Alaska, and they can live for more than 2 years. So they can be hundreds of kilometers across and go all the way out into the Gulf of Alaska. And because they transport water from the shelf offshore, that water has got a lot of iron in it. So when they go out there, you get this nice sort of blooms in these around these eddies from the iron input to the nutrient-rich waters.
Very cool. Yeah, amazing. All right, we're actually getting quite a few questions, aren't we, Deb? So we'll watch our time. We have a couple more minutes, but one of the ones here was also, what is that big orange rusty, talking about iron colored? What are those blooms in bays that are really orangey red color?
That's it's a good question, but I think that a lot of the time they're this Noctiluca scintillans, so the one that bioluminescence, but there are some other species as well. So to really know for sure, you need to take a sample and take a look under the microscope. But I mean, with Noctiluca, you could actually you could get them in a jar. They're really quite big. So if you took a sample in a jar and you just held it up to the light, if you see those round sort of translucent or transparent sacs, that's Noctiluca. Yeah.
Yeah, that answers another question. Someone was wondering, are there some that are visible with the naked eye? So
yep, those
take
ones
a jar and maybe even have your own little handheld micro glass or something.
Yeah, if you had a magnifying glass, you could for sure see Noctiluca, no problem. Yeah.
Cool. Yeah. All right. A couple more, Deb, do you think?
Sure, yeah.
Thank you, Brian.
Whitney asked, how do the sizes of zooplankton compare to the size of phytoplankton? And are any individual phytoplankton visible to the naked eye? Is that the one you just answered?
That's the one we were saying, yes.
Yeah, yeah. So the Noctiluca is big, but it's heterotrophic, so it's not actually photosynthesizing. So technically, it's not really phytoplankton. Well, it is, but like it also isn't because it's not photosynthesizing. But there are bigger phytoplankton. They're the biggest, like the ones that we get here are the diatoms, and they might be like a 1/10 of a millimeter, maybe a big one. So, but they can form chains of lots of them all stuck together, and those can be millimeters long. So you can see those chains with the naked eye. And then someone asked about the zooplankton sizes as well. And the zooplankton can be quite big. I mean, the jellyfish are like outsize exception, but like the krill would be 3 centimeters long, big ones, and then a lot of the copepods would be on the side. So this gets my point of the importance of phytoplankton size. So a lot of the copepods are about 1 to 2 millimeters in size. So you can imagine that, if you have these big long chains of diatoms that are a millimeter or a couple of millimeters long, that's very difficult for a copepod to eat. Like it's this thing that's almost the same length as itself. So diatoms that's why part of the reason why there's so many diatoms, why we get these huge spring blooms, is because it takes bigger organisms to eat them. And so a lot of the small zooplankton are eating actually smaller phytoplankton. Yeah.
Yeah, very cool. All right, here we are. Well, one more, do you think? We're just at almost 9. So
there's one there from Catherine Griffiths.
Yeah, that'd be great.
How does the nutrient load from fresh water runoff impact the phytoplankton production? I'm thinking of human inputs such as fertilizer, sewage, and road runoff.
Well, yeah, so there's that part of it, right? So if we looked at the unimpacted system, then it's what I was referring to previously, that the nitrogen can be very low in those systems, and the phosphate can be quite low as well. But if you go to a human impacted system, one with that's in a farming area, because of fertilizer, they can be high in nitrogen or phosphorus, and so that can make a contribution of nutrients that can stimulate phytoplankton growth. And so we call that that human-induced stimulation. We call that eutrophication. But so it's interesting, like you think you just throw nutrients in the water, stuff will grow, everything will just be, it's actually a good thing. But if you have too much, you end up with low oxygen, like you can get dead zones because of the decomposition. So you have too much growing, a lot of animals, everything that grows, all the phytoplankton will die, it will sink to the seafloor, and then it'll decompose, and then you'll lose oxygen, and that happens in coastal waters. So that's something we have to be really careful of, especially in places that have limited circulation. And then the other part of it is that the ratios of nitrogen phosphates, nitrate phosphate silicates is very important. It affects which phytoplankton are the winners and the losers. And so there are natural ratios out there. And then we disrupt that by putting in our own human input of nutrients. And so that can change the species composition. And then you could get toxic algal blooms, for instance. Like a lot of phosphate from fertilizer can cause toxic cyanobacterial blooms, for instance. So it's a complicated story in a way, but and there's different positive and negative effects that can occur.
I know we're just at time here, but I did want to mention, because a few people asked about the heat dome. And so if you check back to the second letter you got, there was a link there to a talk that Dr. Chris Harley from UBC gave to Nature Vancouver, and he talks specifically about the heat dome. And I'll send that link out again. But I wonder, I know people have to leave. Are you open for one more question, Brian?
I'll take one more question, yes.
Okay. Well, maybe this is one everyone's interested in is, I know there are many changes that each of us need to make to mitigate climate change impacts on the Salish Sea. What is the number one change that you think we should make? And thank you for your excellent presentation.
That's a really difficult question. I often think about the local impacts and then the global impacts. And I think that maybe, there are more immediate local things that we can do. So I think that collectively we could make changes that could really affect the health of the Salish Sea. And while we're trying to do things about the global problem, having a healthier environment here can make the environment more resistant to the effects of climate change. And so doing things like limiting pollution, optimizing marine habitats, looking after all the parts of the system, it's not just about the open water, and it's not just about the kelp, and it's not just about the eelgrass, and it's all those things are connected. And I think one of the things that's so important here is that we have an extreme, really people must understand how extreme our land-sea connection is because of all the rain. And so we have to take care of the land as well when we're thinking about the ocean. And so understanding that and then doing the things that we can to optimize the health of the system is, I think, can just put us in the best position to survive or resist, be resilient against climate change. And so, yeah, pollutions would be one of those things. Looking after nearshore habitats, I think we know all the things we should be doing. We just need to implement them.
Yeah, so true. So true.
Yeah.
Okay, well, thanks so much, Brian. Like Ali said, we really appreciate this talk Wednesday. It just lays the foundation for the whole rest of the program. And there's so much information to chew on and lots to look up, lots to link. Thanks to everyone for your participation, all your questions. I'm sorry we didn't get to all of them. I don't know if you can contact Brian if you have any questions that you just need answered.
I'm happy for people to contact me. I just be warned that I may take a while to respond, so don't hesitate to email me again if you don't get a quick response. Especially this time of year, it's very busy with teaching and finalizing semesters and things. Yeah, but yeah, it was my pleasure to be here, and thanks for all the great questions. I'm sorry we couldn't spend more time answering those, but have a great rest of your evening.
Thanks everyone for participating, and see you next week.
Have a good night.